Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 11 of your OpenStax book, Mechanisms of Microbial Genetics. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for Chapter 11 include being able to describe the process of DNA replication, explain the process of transcription and translation, describe the different types of mutations, and discuss examples of control of gene expression. We begin today by looking at DNA. DNA, in a previously mentioned video, was described as a polymer of nucleotides. The polymer of nucleotides contains a genetic code that is passed from parents to offspring. This genetic code ultimately provides cells with the information needed to construct all of the proteins for the cell. The central dogma describes this relationship. Information in DNA stored in genes is copied to mRNA, and the information coded as sequence of bases is translated to a sequence of amino acids to make proteins. There's another way to look at the central dogma. If we consider a cookbook analogy, we can follow the genetic information from its point of storage to its point of expression. The grandmother's cookbook illustrated in this picture is an example of a set of instructions that one might use to make a wide variety of dishes. Those dishes are analogous to proteins. The grandmother's cookbook is analogous to your DNA being stored in the nucleus of a cell. Genotype and phenotype. The genotype is all the genetic information that is stored in the cell, while the phenotype is what is expressed. So as genes are transcribed and translated, the proteins they produce make something that we can see outwardly. That is your phenotype. DNA replication. DNA replication is the process used to copy DNA. DNA replication in living things is semi-conservative, where the new DNA molecule is composed by opening an old DNA molecule and using it as a template. Subsequently, each new piece of DNA is made up of one old piece of DNA coupled with one new piece of DNA. New nucleotides are added based on the complementarity between the bases as seen on the old strand. The origin of replication. Replication begins at ORIs, where the two DNA strands are separated, opening up a replication bubble. Proteins and enzymes attach in this opening. In prokaryotes, replication proceeds in both directions from each origin until the entire molecule is copied. Gyrase unwinds the DNA. Helicase separates the strands by breaking the hydrogen bonds between the bases. Single-stranded binding proteins keep the strands apart. Here we see an illustration of that process. DNA polymerase and primase. DNA polymerase III adds nucleotides in the 5' to 3' direction on what is referred to as the leading strand. It needs a free hydroxide group to start attaching. Primase makes primers, which are short RNA sequences for the DNA pole to get started. Leading and lagging strands. The leading strand, or template strand, is made continuously in DNA replication. The lagging strand is made in a discontinuous fashion. Small chunks of DNA are made and then have to be assembled. The lagging strand's chunks are referred to as Okazaki fragments. Exonuclease removes the RNA primers on the lagging strand. DNA polymerase I will then fill the gaps with DNA nucleotides, and ligase will seal the gaps. The result is a new DNA molecule. See Table 11.1 in your OpenStax book for a summary of all the enzymes used in this process. Replication in eukaryotes. Eukaryotes contain multiple linear chromosomes, each with multiple replication origins. The mechanism of replication seen in a eukaryotic chromosome is similar to what you've witnessed in the prokaryote. Telomeres and telomerase. Telomeres are special sequences that protect the ends of DNA molecules from erosion. The enzyme telomerase maintains these ends. Rolling circle replication. Some plasmids, bacteriophage, and animal viruses contain circled DNA. Rolling circle replication is a rapid, unidirectional DNA synthesis process illustrated below. Transcription and translation. While the genome contains instructions to make all the proteins for the cell, these instructions are not all produced at all times. Gene expression regulates which genes are to be transcribed and subsequently translated into proteins. Proteins connect genotype with phenotype. Transcription is the process that synthesizes mRNA using DNA as a template, and translation is the process where mRNA is read using rRNA and a protein is assembled using tRNA. RNA polymerase is used for transcription. RNA polymerase synthesizes RNA using antisense strand of the DNA as a template. It adds complementary RNA nucleotides to the 3' end of the growing strand. One major difference between RNA production and DNA production is that RNA polymerase adds uracil in a pairing with adenine instead of thymine. The initiation of transcription. DNA double helix must partially unwind for transcription to begin. A transcription bubble forms. RNA polymerase will then bind to a sequence in the DNA upstream or before the gene, called a promoter. Then this starts transcribing. No primer is needed. Elongation. The elongation phase synthesizes RNA in a 5' to 3' direction. Termination. When RNA polymerase arrives at the terminator sequence, the enzyme stalls. This releases the mRNA strand to undergo post-transcriptional modification. Differences between prokaryotes and eukaryotes. In prokaryotes, genes that are related are often transcribed together, mRNA encoding multiple genes at a time, which is referred to as polycistronic. In eukaryotes, the mRNA is monocistronic, or only a single gene. In eukaryotes, the mRNA is processed, where non-coding sequences are removed and then coding sequences are spliced together. A 5' cap is added for protection, and a 3' poly-A tail is also added for protection. Translation is the synthesis of proteins. This occurs in ribosomes, which are made of rRNA. tRNA and many other proteins participate in the process. mRNA contains the instructions in the form of a codon, or 3 base letter code for amino acids. tRNA must match to the mRNA codon and subsequently present the called for or appropriate amino acid in the sequence. The genetic code is a redundant code and can be seen here in the table at the right. The table illustrates the amino acids that each codon calls for. Triplets of nucleotides code for one amino acid. Those triplets of nucleotides are equivalent to codons in mRNA. A start codon, the only start codon that exists that we know of, is the start codon AUG, which codes for methionine. Stop codons are nonsense codons, or codons that do not call for a specific amino acid. There are 3 illustrated on the table in red. Ribosomes. Ribosomes have a large and a small subunit made of rRNA and proteins. Ribosomes are different in prokaryotes and eukaryotes. In prokaryotes, transcription and translation can happen simultaneously. Transfer RNA. tRNA, as it's sometimes called, is a three-dimensional structure with an amino acid binding site and the anticodon at opposite ends. The anticodon is complementary to the codon of the mRNA. tRNAs are specific to their amino acid. The initiation of translation. Ribosomal subunits, mRNA, the initiation, tRNA, and other factors all assemble to begin the translation process. Elongation. 3 sites in the ribosome, the A, P, and E site, are all used during the elongation phase, or the phase during which the RNA, rRNA, is read and a protein is produced. The A site is where an incoming tRNA amino acid will bind. The P site is where the tRNAs with amino acids that have already formed peptide bonds will sit. And the E site is the exit site where the tRNAs who have released their amino acids will depart. tRNAs with amino acids enter site A and a peptide bond forms between amino acids, then move to site P, the tRNA amino acid bond breaks, and the tRNA exits. Termination. Stop codons do not have complementary tRNAs. Therefore, once reached and read, the polypeptide will be released, and the complex will dissociate. Transcription and translation. In prokaryotes, it is coupled. In eukaryotes, it is not. Transcription occurs in the nucleus, while translation occurs at the ribosome, which might be free-floating in the cytoplasm or attached to the endoplasmic reticulum. Protein targeting, folding, and modification. The removal of translated signal sequences, or short tails of amino acids that aid in the directing of a protein to a particular cellular compartment, occurs shortly after the amino acid sequence has been released. Proper folding of the polypeptide and association of multiple polypeptide subunits facilitated by chaperone proteins can guide the amino acid sequence into becoming a three-dimensional structure. Proteolytic processing of an inactive polypeptide can release an inactive protein component, and various chemical modifications, such as phosphorylation or methylation, can occur adding to an individual amino acid. Mutation. During the process of transcription and translation, it is not unheard of for mistakes to occur. A mutation is any mistake or change in the base sequence of DNA. These mutations may occur in the original triplet code or anywhere along the RNA processing from transcription through translation. They may result in a change in the product encoded by the gene. They may affect the function of the protein. It's important to note that not all mutations are bad, nor do all mutations result in such changes. Agents that result in the production of mutations are referred to as mutagens. This is an example of a point mutation. Point mutations occur as a single base is changed in a DNA strand. Point mutations can be silent, where the amino acid actually remains the same, even though the base has been changed. This is possible because of the redundant nature in our genetic code. There are many amino acids that have redundant or multiple DNA triplets and subsequently RNA codons that call for them. A missense mutation is also an example of a point mutation. In a missense mutation, the base change causes a different amino acid to be called for. And finally, a nonsense mutation may also occur as a point mutation. This results in a stop codon being produced where one should not be or should not belong. Frame shift mutations. Frame shift mutations involve the insertion or deletion of one or two bases. They literally shift the reading frame of the DNA or RNA strands produced. An illustration of how this occurs can be seen below. All the amino acids after the mutated portion of DNA will be affected and non-functional. The cause of mutations. Spontaneous mutations are rare, but they do happen. Spontaneous mutations occur at a rate of 1 to the 10 to the 9th power base pairings. Chemical mutagens are more often the cause of mutation than spontaneous mutations. Chemical mutagens are often carcinogens. Things such as nitrous acid cause base pair changes at the adenine-thymine, guanine-cytosine unions. Base analogs can form from chemical mutagens, where we see or insert structurally similar molecules but different molecules than the normal nitrogenous bases. Intercalating agents can also be inserted into the DNA and cause frame shift mutations. Ultraviolet light, X-rays, and gamma rays are also mutagens. Ultraviolet light causes bonding between adjacent thymines, referred to as thymine dimers. X-rays and gamma rays cause the formation of ions and free radicals that react with DNA, causing base substitutions or breakage of sugar phosphate backbones. Nonetheless, your DNA can be repaired. A DNA molecule is not always replicated correctly, but DNA polymerase does proofread its work and can fix mistakes that have been made. Mismatch repair reverses the synthesis reaction to remove a mismatched nucleotide, then resuming synthesis in the forward direction. Bacteria can repair thymine dimers using nucleotide excision and photoreactivation. The Ames Test. The Ames Test is a rapid screening for carcinogenic potential. Carcinogens are mutagens, and this test measures the ability of a compound to induce mutations. Auxotrophs are organisms that have lost the function due to mutation. They cannot synthesize a specific amino acid. Horizontal gene transfer. Horizontal gene transfer involves sexual reproduction and can provide genetic diversity. Asexual reproducing organisms can gain genetic diversity by horizontal gene transfer. 3 mechanisms can occur in bacteria: transformation, transduction, and conjugation. This is an example of transformation. In transformation, the cell takes up DNA directly from the environment. The DNA may remain in a separate plasmid or be incorporated into the host genome. Once incorporated into the host genome, it becomes recombinant DNA. Transduction occurs when a virus injects its genetic material into a host. Phages, or viruses that infect DNA, inject DNA that is a hybrid of viral DNA and DNA from a previously infected bacterial cell. This can be generalized or specialized. Lysogenic conversion often follows this injection. Conjugation is sexual reproduction of sorts between bacteria. F plasmids code for a conjugation pilus, as illustrated to the right. The cell that donates genetic material is referred to as the F plus cell. The cell that receives genetic material is the F minus cell. There are no genders in bacteria. DNA is transferred between the cells through a cytoplasmic bridge after a conjugation pilus draws the 2 cells close enough to form said bridge. High frequency of recombination cells. HFR cells are cells where the F plasma can occasionally integrate into the bacterial chromosome. The imprecise excision of the F plasmid from the chromosome of an HFR cell may lead to the production of an F+ plasmid that carries chromosomal DNA adjacent to the integration site. This F+ plasmid can be transferred to an F- cell by conjugation and can incur or transfer things such as antibiotic resistance genes. This is an illustration of how that process might play out in nature. Transposons. Transposons are also known as jumping genes. They are small segments of DNA that can move from one region of a DNA molecule to another. They contain transposition genes and may contain another gene along with it. They may insert into genes and potentially inactivate them, as illustrated below. Gene expression. Gene expression is all about what we see when the genetic material contained in a gene is transcribed and translated into protein, but it must also be regulated. Regulation of which genes are expressed and how will be explored as we examine gene expression. In prokaryotes, gene expression is controlled mostly at a transcriptional level. In eukaryotes, it can be controlled both transcriptionally and post-transcriptionally. This is an example of an Operon and prokaryotic gene regulation. Here we can see structural genes together with those that are coding for enzymes involved in the same pathway. There is a single promoter and a regulatory gene that might code for a repressor or activator, which binds to the operating gene. This controls the transcription of the gene. As an example, we have the tryptophan operon. The genes are involved in the production of tryptophan, and it is controlled by repression through the use of a repressible operon. If tryptophan is present, the repressor will be bound to the operator and no transcription will occur. If tryp is present, then the repressor goes away and transcription can also occur. Here we have the lactose operon, which is an inducible operon. It involves enzymes needed to degrade lactose. The lactose allolactose is an inducer which binds to the repressor, which cannot bind to the operator. It can be regulated by your glucose levels. When glucose is low, cyclic AMP activates the operon. When glucose is high, it inhibits it. This is a graph illustrating the growth of E. coli in both glucose and lactose. You can see by studying the graph that E. coli prefers glucose. Once used, lactose induces the operon, and the enzymes needed to use lactose are transcribed and lactose can be used. The global responses of prokaryotes. Alarmones are things that we see in the global responses of prokaryotes. They are small intracellular nucleotide derivatives. They change which genes are expressed and can stimulate the expression of specific stress response genes. Attenuation and riboswitches also fall into this category. They can control coupling of transcription and translation. Enhancers can participate in things like epigenetic regulation or the methylation of DNA to make it less accessible. This brings our coverage of Chapter 11, the mechanisms of microbial genetics, to a close.